Most important things, though, is that we make sure before we do any of those actions that we've really thought through those. And we want to be proactive on that process. We don't want to have something come out of the lab in 2 years and then go, all right, now we have a 2-year process of thinking through the appropriateness of that action. We want to think through the appropriateness of those actions once we understand them, even if they're not fully ripe for implementation at that point in time. And so a year ago, NOAA thought about this, trying to think proactively, and said, what group would be the best one to give an independent review to these actions, to all these big ideas that are being considered? What group can do that for us? And the immediate answer to that, within the US at least, is the National Academies of Science, an independent organization that reviews science in all fields, whether it's engineering, biology, medicine, and helps provide consensus-based recommendations from experts that serve on panels that are the best of the best in the various fields. And so NOAA commissioned the National Academies of Science to do a review on coral interventions. They've already started that process. They're about 1 year into that. They've got their initial report, which kind of looks at and explains the various actions that they're considering. Over the course of this next year, they're going to be taking a deeper dive into those actions, thinking about how to evaluate the risk of those actions, and eventually proposing a decision support tool for managers to use in evaluating when and how to use those actions on their reef. And so to talk about that report, we've got the chair of the panel, Dr. Stephen Palumbi. Steve is a professor at Stanford University. He's considered a genetic detective in many ways, having looked at how to identify whales in retail markets, shark fin soup, and where conch comes from around the world, as well as being one of the leading experts on coral reef genetics. Thanks, Steve.
Thank you very much, Tom. Thank you very much, Tom. As Tom said, I had the enormous honor of leading this committee to put together the ideas that have come from you about coral reef resilience around the world. This is the list of the committee. Some members are here. Ken Anthony is here. Andrew Baker is here. Bob Richmond is here. It also really was incredibly powered by the staff of the National Academy of Sciences. Emily Twigg in particular is here, who led us, who told us what to do, and honestly, this is a great committee because they actually did it, and that resulted in what we have today. What I'm going to try to do today is to go through some of those interventions, and I find the task has already been largely done. Over the last couple of days, we've heard about those interventions either in summary by our colleagues in Australia, but a lot of you are already doing them or thinking about them or incorporating them into what it is you're thinking. Sarah just told us what the context is. Things are changing. This is one of my favorite corals in Samoa, AH93. I followed this coral since it was a little nubbin growing up over a couple of years, and it had gotten nailed by the 2015 bleaching. This is the context that we're all working in. The first thing, though, about these interventions that we're going to talk about is that they do not live alone. And particularly when we're thinking about the dangers of climate change, that it's incredibly important to realize that climate mitigation, reducing CO2 into the atmosphere, not only stopping putting it in, but having it be reduced is in fact existential for the existence of coral reefs through the next century. It all pivots on that. In addition, we all know that there are local stressors. There are things we're doing to coral reefs that have them create decline that have nothing to do with climate change. They're also critically important to work with. But even if we had a handle on those, both of those, it wouldn't be enough because the climate is already changing. Bleaching events are happening more and more and more around the world, and they're affecting coral reefs in a huge way. So our view of these interventions is that because the climate is on a path to change, even if we get a handle on CO2, we must have the ability to increase coral resilience, at least in our toolbox. This is a toolbox of what could be done. The committee broke down these different interventions from the literature, from the peer-reviewed literature largely, talked about what it is, the benefits and goals, how to do it, the risks, the limitations, the infrastructure needed. And we talked about resilience and persistence. Resilience being the idea of generating more stress tolerance, the idea of the cover of the coral versus the function of the ecosystem, and the persistence of the reefs into the future. We had a series of public workshops, webinars. Some of you were very important participants in that. What we have gathered together from the literature and from the practitioners is essentially 4 intervention categories. We have heard about a bunch of them already. We can talk about the genetics and reproductive interventions. We can talk about physiological interventions, coral population and community ones, and then environmental interventions. And what I want to do is to walk through some of those. I'll give some examples of those so you see the kinds of things that have come out in the literature. I'm not going to do this just walk through every single one. If you don't mind, just me telling you 23 things in a list is not going to be all that engaging. So we'll just give you some examples. Genetic and reproductive interventions are things like managed selection, managed breedings to increase the number of corals on reefs, standard, if you will, restoration, but also increase the resilience of those corals to climate effects. So it's not just restoration, but restoration with particular genotypes. And they have a number of different ways. There's a number of different ways of going about that. This is some work that we have been doing in my research project in Palau, essentially testing individual corals for their heat resistance phenotypes. That little tank there on the lower right is our stress tank. It's built small enough so it fits in the overhead compartment of a jet. Everything you need to run it is inside it, and you can use it to heat stress corals. On the right there is a set of corals that bleach under the conditions that we're running in the tank. The coral next to the right did not bleach. The coral to the left did bleach. They're growing on the same reefs. And this is what we almost always see. There are differences in the ability of corals to resist heat stress. We've been doing this for about 400 corals of Acropora hyacinthus around Palau, and this just shows you one of the first ever maps of where you can find heat-resistant corals in an archipelago. This is the sort of thing that is being able to be done all over the world to generate the raw material, if you will, for using heat-resilient or heat-resistant corals in future restorations. You've seen these sort of slides over the last couple of days. The ability to restore reefs with hundreds of or thousands or tens of thousands of corals allows the greater persistence of those reefs, allows the maintenance of the ecosystem services that they provide. And even if you're not managing them for resilience, it adds greatly to the persistence of those reefs. There have been trials of different kinds of managed breeding already. This is one. This comes from Grover Dixon's work in Misha Metz's lab on the Australian Great Barrier Reef, also working with Lena Bay, where corals from the northern and the middle of the Great Barrier Reef were brought together, crossed. The larvae were tested for their heat resistance with a simple result that, well, the northern corals were more heat resistant, and so were their offspring when they were crossed with corals from further to the south. It's just one example of the ability to take corals from the wild and use managed breeding techniques that are very common in, say, selective breeding in other animals and plants to work on increased resilience of these animals. Another example from this genetic and reproductive category is potential outcrossing. This is a recent paper out of Madeleine Van Oppen's group, taking 4 different species of Acropora, crossing them, creating interspecific hybrids, and testing them for their ability to grow in different temperatures and at different speeds. This is just the beginning of this kind of work, and we're just seeing the first lab tests and some of the first field deployments of some of these. Last in this category are things like genetic manipulation. Genetic genetic engineering of the coral or the symbiont. Right now, genetic manipulation of the symbiont is not technically feasible, but over the last couple of months, last couple of years, techniques to genetically engineer corals have moved along in a strong fashion. This is a paper that appeared a few months ago by Phil Cleves and his collaborators, Lena and Misha Matz and colleagues at Stanford. And what they were doing is using CRISPR-Cas9 injected into coral larvae to genetically manipulate those larvae. There was one set of phenotypes that they observed in this very first paper. The top image there is a normal coral larva taken in fluorescent lighting that glows because of the fluorescent proteins. If you inject CRISPR-Cas9 that's designed to go in and knock that gene out, then what you get is larvae that don't fluoresce, and that's the panel D. The lack of any image there means that that gene has been successfully knocked out, the first genetic manipulation of corals. So these technologies are growing. They're not even in the lab in terms of making resilient corals, but they're beginning to be worked on in terms of the methodology. We brought these interventions together into a relatively large table. That's 3 pages. This is just 1 page of it, showing the different interventions, what it is, the feasibility, the scale, et cetera, as a way of sort of one-stop shopping, if you will, for interventions. Many of these are only being done in the laboratory right now. Some of them are done in field trials. Some of them are not feasible at all, like genetic manipulation of the symbiont. So we tried to characterize where they are right now. And one of the interesting things about this is that the examples that I showed you are largely from papers that have been published over the last 3 to 6 to 12 months. The amount of energy going into this is amazing. The rapidity with which we are learning is amazing. So you should keep up with what's going on. In fact, when we would have different meetings on this topic, the first thing out was like, okay, what came out of the last 2 months that we have to take note of, because the field is moving so fast. There are physiological interventions that are different than genetic and reproductive. That's improving the health of corals, changing their physiology. In this case, we're not just talking about the coral, but also talking about the symbiont and the microbiome. To give an example, pre-exposure, taking corals, exposing them to, say, slight increases in heat gives them acclimatory changes that allows them to deal with more exposure to heat later. But it's temporary. And if you bring them in cooler water, then that disappears. So that kind of intervention might be possible, but we don't know how long it would last. Manipulating algae is something that is much more familiar to coral biologists in the room. This is just an example from Silverstein et al. In 2015, effectively taking corals, bleaching them, swapping out different kinds of Simiodiniaceae that have a different stress tolerance, and seeing that improves the stress tolerance of that holobiont. Again, those tend to be temporary, longer time periods than just, say, acclimation, but we don't really know the dynamics too well after a couple of months. Active research is going on right now. A different kind of manipulation that is a little less familiar is the manipulation of the microbial community, say, with probiotics. This is a paper that's just been published by Rosado et al., in which they took Poecilopeira damicornis, isolated bacteria, screened them, identified them, cultured some of them, put them together in a slurry, picked the ones they thought might be beneficial. Thought is the important word, we're guessing, and then added those to as probiotic slurries back to the corals. And then follow them over time. This is, as far as we know, one of the first applications of probiotics to corals. It's obviously not ready to be used on a reef scale. We don't know how it would work on any bigger scale than just the lab. Nevertheless, it shows what action is going on and what people are trying to do in the lab. Again, another table of what those different manipulations are. In this sort of category, including are things like using antioxidants or nutrient supplements to try to overall increase the physiological health of corals. Coral population and community interventions include essentially moving corals that are adapted to different environments into new environments. And although it's just one kind of intervention, we broke it up into 3 categories of increasing distance because of the increasing cost and risk as distance goes up. So assisted gene flow is moving populations of corals around within their range to take resistant genotypes and moving them into populations that are slightly less resistant. Assisted migration is moving corals just outside their current range. And introduction to new areas would be moving them from ocean basin to ocean basin. Assisted gene flow is something that's just begun to be done. I'm going to give you an example here from work that we've been doing in American Samoa in Ofu. This is work done in her PhD dissertation by Megan Morikawa, who moved corals from these 2 regions of this island in American Samoa, a hot and a warm back reef pool, to a separate site. She characterized these corals as being heat resistant or heat sensitive based upon the phenotyping from things like those coral stress tanks, and started growing them up in an experimental nursery. This is a view of that. What is circled in red here are the nubbins, the nursery products that came from parent corals that were tested to be heat resistant. What's circled in brown and blue are nubbins from parents of corals in the reef that were shown to be heat sensitive. They were growing for about 8 months before the 2015 bleaching event hit, and what this work showed was that if you essentially set up an experimental nursery with a wide range of corals that have different types of heat tolerance, that in fact the nursery itself reflects the heat tolerance of those parents. They bleached about 2 to 3 times less, those nubbins, when they came from heat tolerant parents than when they came from heat resistant ones. It shows that the restoration technology, when coupled with an idea of looking for resilience in the coral reefs, might be able to lead to a generation of more heat-resistant nurseries. And Megan's here, is going to talk later, and be happy to talk about that work, I'm sure, on 4 species. So these are the sorts of interventions that we've got there. We have to notice that interventions that move corals outside their range and interventions that move corals to other ocean basins have not been done. Sarah was just talking about those as being reasonable, risky categories, and that there are no examples of those being done in the field. We don't know anything to say about the risks there, but the risks of doing that must go up with the distance that we move those corals. There are environmental interventions as the last category. We heard a talk about this a couple of days ago in this room, that is changing the environment, not changing the corals, and then allowing that environment to be more benign to corals during bleaching events. Examples of shading, possibly cloud brightening, which we heard about a couple days ago, shading the corals with regular shades or with monomolecular layers, all to reduce the amount of light during heat events. But there are also interventions, say, of cooling reef water, of changing the way that the reefs are allowed to heat up by either pumping water up or producing, say, a grid of cool water pipes along them. So this is the category here of these different interventions. We also included a couple of interventions that were not heat-based. They were ocean acidification-based, based upon efforts to reduce CO2 in seawater, either chemically or through using seagrass beds or algal beds to pull down CO2 during the day. Interventions range in their feasibility, and they also range in where they have been deployed. The red here on these diagrams are things that are not feasible, have never been deployed. Yellow tends to be things that are just deployed in the lab. Green are things that are deployed in the lab and maybe in field trials. Blue are things that are globally successful. There's no blue, because these interventions are really moving into the testing phase, and these resilience interventions really have not been developed that are known to be applicable on large regional or global scales. So that gives you a sort of summary of the broad sweep of this. There are 23 interventions there. The report is relatively long because it goes through every single one of them. At this point, I have to say the committee that we were working with is a fabulously amazing group. None of them were experts in all of these interventions. And so we had to take them bit by bit and teach one another about them. Some of those interventions, people in the room here are much more expert at than we are. So by all means, consider this an opening for a dialogue about what's new, what's going on, how these things are being developed. At this point, the report shows what it is we know now. And it represents a toolbox. It represents what we could do. And the energy that we saw on this stage an hour ago with people talking about what they can do for restoration is, I think, infectious. It's the kind of thing that we know we have to do. Sarah was completely right that looking at the future means that doing these sorts of things. What I think the committee and me personally found just the most amazing is that when you look in the toolbox for what to do about increasing resilience of corals, it is not empty. It's got a lot in it. Now, we don't know exactly how to use all those tools, mind you. Some of them are still wrapped in plastic, and you can't really even read the instructions. But they're there, and that represents a huge opportunity in the future. A lot of risk, like Sarah said, yes. But at this point, it means that we have things that we can do. The report is out. It's downloadable on that website. The National Academy group is now moving to trying to figure out decision-making pathways for using these interventions. But at this point, I have thanked the committee, I've thanked the National Academy, but what I really have to do is thank you all. The amount of energy and enthusiasm that's been over here in the last few days has been, amazing and infectious. Tali and Tom have put together an incredible group that really shows, more than almost any other coral meeting I have been at in the last 10 or 15 years, that there's ways forward, there's energy, there's optimism, and there's a huge amount of work. So that's where we're going to leave it, with optimism and work. Thanks.